Hi, my name is Dr. Elizabeth Sokol, and I'm one of the registrars in chemical pathology and metabolic medicine at Imperial. We're going to talk about the basics of enzymology. We will go through the protein structure, we'll talk about enzyme nomenclature, and we'll talk about key definitions, enzyme kinetics, and Michaelis-Menten equation. So, first of all, what is an enzyme? Enzyme is a biological catalyst which allows the reactions to take place at a much faster rate without the enzyme being used up in the process. Majority of enzymes are proteins, but there are some enzymes that can be catalytic RNAs. And how does the enzyme does its job is dependent on its structure. So as you can see on this slide, we will talk about the primary, secondary, tertiary, and quaternary structure of the proteins. And what the primary structure refers to is the linear sequence of amino acids in the polypeptide chain. The sequence is determined by the gene encoding the enzyme. Each amino acid is linked to the next by a peptide bond, and it dictates enzyme's overall shape and function. And if there is any mutation or change in the sequence, it can affect enzyme activity. As you can see in the right top corner of the slide, the secondary structure of enzyme refers to the localized folding patterns within the polypeptide chain, and they're stabilized by the hydrogen bonds. So you can see two common forms, the alpha helix, which is a spiral-shaped coil, and a beta sheet, which is a folded sheet-like arrangement. They are formed by the hydrogen bonding between the backbone atoms, but note it's not the side chains. And these structures help determine how the enzyme folds into its final 3D shape, which is its tertiary structure that you can see in the lower left corner of the slide. The tertiary structure of an enzyme is an overall 3D shape of a single polypeptide chain. It results from interactions between the side chains, the R groups of amino acids, and the key interactions that stabilize the tertiary structure of the protein are hydrogen bonds, ionic bonds, disulfide bridges, which are the covalent bonds between cysteine residues, and the hydrophobic interactions between the nonpolar groups. This level of structure determines the active site of the enzyme, where the substrate binds, as well as the specificity of the enzyme for its substrate. And it helps determine the overall function of the enzyme. As you can see in the bottom right corner, the quaternary structure of the protein, which consists of two subunits. On this picture, there is a subunit 1 and subunit 2, and it refers to the arrangement of multiple polypeptide chains, subunits, in a functional enzyme. Not all enzymes have a quaternary structure, only those that are made of several subunits. And the key features of the quaternary structure is that each subunit may have its own active site or can help in the overall function, and that the subunits are held together by the same types of interactions as in tertiary structure. And one of the examples, as you know, is hemoglobin, which consists of the 4 subunits. Now we will talk about the two key models that have been proposed to describe the function of the enzyme. As you know from your A levels, one of them is a lock and key model of the enzyme action, and it was proposed by Emil Fischer in 1894, and it's one of the earliest explanations for enzyme specificity. And what it tells is that only the correct substrate fits exactly into the enzyme's active site, like a key fits a specific lock. And once bound, the enzyme catalyzes the reaction and then releases the product. It is highly specific, so each enzyme only fits one or a few substrates, and no major changes occur in the enzyme structure during the binding, unlike in the induced fit model. And in the induced fit model, which you can see where the keys sort of line in a mold, and this model has been proposed by Daniel Koshland in the 1950s, it shows that the enzyme is not rigid. The enzyme is flexible and can adapt to fit the substrate. So the substrate binds to the enzyme's active site. The binding causes the enzyme to change shape slightly, just enough to create a perfect fit, and the induced fit improves binding and helps catalyze the reaction. So to give an example, substrate approaches the enzyme, enzyme's active site shifts shape to fit the substrate, the reaction occurs, which lowers activation energy, and the product is released, and enzyme returns to its original shape. On this slide, we are going to talk about enzyme commission nomenclature. So there are many enzymes that exist, and Enzyme Commission decided to create a standardized system to help classify and name enzymes based on the reactions that they catalyze. Enzyme Commission is a part of International Union of Biochemistry and Molecular Biology, and you can see in the bottom right corner a link for their website where you can see a catalog of all the enzymes and you can find a more detailed information. But if you can see in the right upper corner, you have an EC and numbers 1, 2, 3, 4. And what it says is that each enzyme is assigned a unique EC number in the format that I've just described. And each number has a specific meaning. So for instance, number 1 shows to which of the 6 main divisions or classes the enzyme belongs. And actually in 2018, they've added another class. So there are actually now 7 main divisions or classes. Number 2 indicates the subclass. Number 3 gives the subclass. And number 4 is the serial number of the enzyme in its subclass. So the 7 classes that I mentioned, with class 7 being the newest one, which is called translocases. You can see in this slide that you have different enzymes that catalyze reactions using different reaction types such as oxidoreductases, transferases, hydrolases, lyases, isomerases. Now we will talk about some key definitions that often come up in the exam. And first, let's talk about isoenzymes. So isoenzymes are different molecular forms of the same enzyme. They catalyze the same reaction, but they differ in their physical properties, such as their amino acid sequence, kinetic properties in terms of how fast they work, their regulation, the tissue distribution, and their physiochemical properties, such as electrophoretic mobility, resistance to inactivation, solubility, and catalytic characteristics. And the examples you can see is LDH and ALP. So for instance, lactate dehydrogenase catalyzes the conversion between lactate and pyruvate in glycolysis and aerobic respiration, and it has 5 isoenzymes, LDH1, LDH2, LDH3, LDH5, and they are made from different combinations of 2 subunits. I will show this on a subsequent slide in more detail. Isoform is a molecular form of the enzyme, which underwent post-translational modifications, and they usually have similar catalytic properties. Allozymes are isoenzymes that are derived from different alleles of the same gene. On this slide, you can see the two different genes that encode LDH. So you have LDHB gene and LDHA gene, and you have mRNA, and there are two different proteins that are formed that form H subunit and M subunit. And as you can see, different combinations of subunits can give rise to different isoenzymes, 5 isoenzymes of LDH. So for instance, LDH1 in the top right corner is made of 4 H subunits. LDH2 is made of 3 H subunits and 1 M subunit. LDH3 is made of 2 H subunits and 2 M subunits. LDH4 is made of 1 H subunit and 3 M subunits, and LDH5 is made of 5 M subunits. Why is this important? Is because isoenzymes reflect the expression of different genes or gene variants. And during physiological changes, such as during development, stress, or disease, the relative activity of these genes shifts, altering the isoenzyme profile in tissues. When a normal cell becomes cancerous, for instance, there is often a reversion to fetal embryonic gene expression patterns, and this leads to reappearance of fetal isoenzymes that were suppressed in adult tissues, for instance, placental ALP. Or it can be used as a tumor marker. For instance, cancer cells rely heavily on anaerobic glycolysis, even in the presence of oxygen, known as the Warburg effect. And LDH isoenzymes shift towards LDH5, which supports lactate production from pyruvate. And the certain tumors reactivate genes that were only active during development, leading to fetal isoenzyme expression, which is very important in terms of diagnostics. So, enzyme activity, how do we assess it? Enzyme activity refers to the rate at which an enzyme catalyzes a reaction. Essentially, it tells you how fast it converts substrate into a product. It reflects how efficiently an enzyme is working under specific conditions. It's usually measured at the amount of product formed or substrate used per unit time. So you can see here the two common units that are used to describe enzyme activity: international unit and kcal. The most common unit used is an international unit, and it is the amount of enzyme that catalyzes the conversion of 1 micromole of substrate per minute under specific conditions, for example, pH or temperature. Whereas kcal is the enzyme activity expressed as 1 mole per second, and it's very large and is not used that often. On this slide, we are going to talk about holoenzyme. So you can see here that a holoenzyme, it's a protein part called apoenzyme. This part is the enzyme itself, which has the active site and is responsible for catalysis. It alone cannot catalyze the reaction without the cofactor. And cofactor is a non-protein component, which can be either a coenzyme or cofactor, which could be a metaline, or it could be a vitamin. And it's necessary for the enzyme to function properly. Together, the whole enzyme is fully active. And the non-protein component, cofactor coenzyme, often helps with electron transfer, group transfer, or stabilizing enzyme substrate interactions. So how do we go from an enzyme to and substrate to product? So enzyme, as we said, is a biological catalyst that speeds up chemical reactions without being consumed in the process. And as I mentioned already, that it works by lowering the activation energy required for reaction to occur. The substrate is the molecule that binds to the enzyme and is transformed in the reaction. Each enzyme is highly specific to its substrate due to the structure of its active site. The product is the result of the enzyme-catalyzed reaction. Once the enzyme acts on the substrate, it releases the product and is ready to catalyze another reaction. The enzyme active site binds to the substrate, forming an enzyme-substrate complex. And this is where the substrate undergoes a transformation. So enzyme catalyzes the conversion of the substrate to the product, and this may involve either breaking a bond, such as in hydrolysis reaction, forming new bonds, such as in condensation reactions, shifting electrons, moving atoms or groups. Formation of product. The substrate is converted to the product, and the enzyme releases the product. And after the reaction, the enzyme returns to its original state and is ready for the next reaction. So what do I mean when I say that the enzyme lowers the activation energy of the reaction? So you can see on the graph here that you've got a substrate and you've got a product, and there is a change in the free energy of reaction, which is shown as Gibbs free energy. And it's the energy in the system that's available to do the work. And for the chemical reaction, delta G represents the difference in free energy between the products and the reactants. Enzymes do not change the free energy of the reaction. They simply lower the activation energy, making the reaction occur faster. So you can see here there is a human trying to push a stone up the hill. And what the enzyme does, it just decreases the slope of the hill and the height of the hill, making it easier for you to push. So less energy is required for you to get to the top of the mountain. And this is basically what the enzyme does. So it provides an alternative pathway with a lower activation energy, but the difference in free energy between reactants and products remains the same. So this is another graph that basically covers the same concept. So you can see on the x-axis there is a reaction coordinate, and on the y-axis there is a free energy, Gibbs energy. So what affects the rate of enzyme catalyzed reactions? So enzyme concentration, substrate concentration, pH, so each enzyme has an optimal pH, temperature, activity increases with temperature up to an optimum temperature, beyond which the enzyme becomes denatured and doesn't work, as well as presence of inhibitors, activators, coenzymes, prophetic groups, and we'll talk about it in the future slides. So Michaelis-Menten curve. So let's talk about some important definitions first. So Michaelis constant, Km, as you can see here, this is the equation, K minus 1 plus K2 divided by K1. It's a substrate concentration at which the reaction proceeds at half of its maximum velocity. It's a measure of the affinity of the enzyme for the substrate. So the lower the Km indicates higher affinity, meaning that the enzyme binds more tightly to the substrate. So as you can see on the top, you've got an enzyme and a substrate, and you've got a forward reaction to the formation of enzyme substrate complex, K1, and we've got a reverse reaction of K minus 1, which goes back to enzyme and substrate. And then you've got a K2, which shows you it's a rate constant for breakdown of enzyme substrate complex to enzyme end product. So K1 is the rate constant of association of ES complex, the forward reaction. K minus 1 is the rate constant for the dissociation of ES complex, so the backward reaction. And Km, as I mentioned, is K minus 1 plus K2 divided by K1. So on the graph, you can see on the x-axis, there is a substrate concentration, moles per liters times 10 to the power of minus 4, and then a velocity, which is an arbitrary unit on the y-axis. You can see red dots on the graph, and you can see that at the beginning, the reaction follows a first order kinetics. So the proportion of the substrate concentration and the enzyme. So as they increase, the rate of the reaction increases. But once you reach a state where each enzyme is saturated with the substrate, the reaction starts to follow a zero order kinetics and you reach a Vmax. The point, so where the half of the maximum velocity, you can see in the red Km. So this is a Michaelis-Menten constant. And this is really important for the purpose of your exams. So as I've said, it's a measure of the affinity, so how tightly an enzyme binds to the substrate. So the lower the Km value, the higher the affinity. So Vmax. So as you can see here, each enzyme is fully saturated with a substrate. So adding more substrate is not going to increase the rate of the reaction, hence Vmax. So Lion-Weaver-Burk plot it helps to show you how the reaction rate changes depending on the enzyme or substrate concentration and the velocity. And you can convert it into a linear graph where you can see in the red box, so 1 divided by velocity equals Km divided by Vmax times 1 divided by concentration of substrate plus 1 divided by Vmax. And this follows a similar linear graph y equals ax plus b, if you remember from your A-level math. And velocity is equal Vmax of substrate divided by Km plus substrate concentration. So as you can see on the graph, the x-axis is 1 divided by substrate concentration times 10 to the power of minus 4, and the y-axis is 1 divided by v, where you the graph intercepts the y-axis. This shows you 1 divided by Vmax. And where the graph intercepts the x-axis, it shows you the -1 divided by Km. So it can help you calculate the Km value and the maximum velocity value. So again, V is the reaction rate. Vmax is the maximum rate when all enzyme active sites are occupied by substrate. And S is the substrate concentration. Km is the Michaelis constant, so the substrate concentration at which the reaction rate is half of maximum velocity. If we look at this equation again, so if we try to look at it as y equals ax plus b, where a is the slope of the line and b is the y-intercept, so where the line crosses the y-axis. And you can see that the slope of the line would be Km divided by Vmax. And again, this is important for the exam, and it helps you determine the values. Now we will talk about enzyme kinetics and what the inhibitors do to the reaction rate. So on this graph, you can see on this diagram the different types of inhibitors: competitive, non-competitive, and non-competitive inhibitors. And in blue, you can see the normal binding, where a substrate just binds to the active site of the enzyme. So what's the difference between competitive, non-competitive, and non-competitive inhibition? I will talk about this in subsequent slides, but this is just a summary slide. So let's talk about the competitive inhibition. So in competitive inhibition, the inhibitor competes with the substrate for the enzyme's active site, and it increases the apparent Km, but doesn't affect Vmax. So let's look at this reaction. So on the top graph, you have an enzyme, you've got an substrate, you have enzyme substrate complex, and then you have enzyme and the end product. And you can see that the enzyme competes with the inhibitor competes with the substrate for the enzyme, and you can have an enzyme inhibitor complex. So the inhibitor is structurally similar to the substrate. And you can see here on the graph what effect it does on the kinetics. So the effect of a competitive inhibitor, it competes with a substrate and it increases the Km value because you need more substrate to reach half of the maximum velocity. But it doesn't affect the maximum velocity because hypothetically the inhibitor can be outcompeted by high substrate concentrations. So you can add so much substrate that you could hypothetically reach a state where each enzyme site is bound by a substrate. Hence, you can achieve the maximum velocity. But you would need a much higher concentration to do that. That's why your Km value changes. So the slope of the Lineweaver-Burk plot is given by Km over Vmax. For a competitive inhibitor, the slope increases because Km increases the effective concentration of the substrate needed to reach half of Vmax. is higher. And you can see here on the y-intercept, the y-intercept is 1 divided by Vmax. Since Vmax is unchanged, in the presence of a competitive inhibitor, the y-intercept remains the same. X-intercept is minus 1 divided by Km. Since Km increases in the presence of a competitive inhibitor, the x-intercept moves closer to 0, so it's less negative. And this is very important to understand. On this slide, we are going to talk about non-competitive inhibitors. So in non-competitive inhibition, the inhibitor binds to an allosteric site, not the active site, causing a decrease in Vmax without changing Km. So a non-competitive inhibitor binds to an enzyme at a site different from the active site, and its binding does not directly compete with the substrate for the active site. This leads to a decrease in the maximum reaction velocity, Vmax, but the Michaelis-Menten constant remains unchanged. A non-competitive inhibitor affects the enzyme's activity by reducing the total number of functional enzyme molecules available for the reaction, thereby lowering Vmax. However, the substrate can still bind to the enzyme, and the affinity of the enzyme to the substrate, represented by Km, remains the same. So if you look at the graph, in the right corner, you can see, again, the slope in the presence of a non-competitive inhibitor increases because your Vmax decreases. So 1 over Vmax value would be higher, while Km remains constant. X-intercept is minus 1 divided by Km. Since Km remains the same, the x-intercept does not change in the presence of a non-competitive inhibitor. Now we will talk about uncompetitive inhibition. An uncompetitive inhibitor binds only to the enzyme substrate complex. The inhibitor binds to the enzyme after the substrate has already bound to it. This type of inhibitor does not bind to the free enzyme. The binding of the uncompetitive inhibitor lowers both the apparent maximum velocity Vmax and affinity for the substrate, leading to a decrease in the apparent Km. Importantly, because the inhibitor affects the enzyme substrate complex, the relationship between substrate and the enzyme is shown on the graph here. So what you can see that Km decreases because the concentration of the active enzyme substrate complex is reduced, and there is a lower apparent Km because the enzyme substrate complex is stabilized by the inhibitor. Apparent Vmax decreases because the inhibitor is only active when the substrate is bound to the enzyme. The overall concentration of the active enzyme substrate complex decreases, leading to lower Vmax. However, this doesn't affect the binding affinity of the enzyme for the substrate at low concentration. Km decreases. Substrate concentration and reaction velocity changes. So on the graph here, you can see that the line has been shifted to the left because both Km and Vmax decrease in the same proportion. X-intercept moves to the right because Km decreases, and the y-intercept increases because Vmax decreases. So this graph summarizes different types of inhibitors and how they would affect the Michaelis-Menten curve and the Wien-Weaver-Burk plot. So in competitive inhibitor, your Km value increases because you need a higher substrate concentration to reach half of the maximum velocity, but you're still able to reach the maximum velocity. And this is shown by the bottom left graph. And you can see the Wien-Weaver plot on the top, where you can see that -1 over Km becomes less negative, and that's why it's shifted to the right, the x-intercept, whereas y-intercept stays the same. In the second graph, at the bottom, you can see that both Vmax is decreases, whereas Km remains the same. And you can see on the top the Wien-Weaver-Burk plot for non-competitive inhibitor. And in the third column, you can see the uncompetitive inhibitor. And you can see that both the Km value and the Vmax decrease. And this causes the curve to shift. And this is the end of the enzymology lecture.